Good afternoon. So today we are going to start with the uh, new topic, that is HMP shunt pathway. Before that, we are going to just review the few things related to carbohydrate metabolism. So if you remember carbohydrate metabolism, we have started with different pathways. So initially we have discussed about glycolysis. So glycolysis, it is also called as Embden-Mayer of Parnell's pathway. So the oxidation of glucose to pyruvate and to lactate we have discussed over there. Then we have discussed about citric acid cycle, which is also called as Krebs cycle or tricarboxylic acid cycle. There we have discussed about the oxidation of acetyl coenzyme A to carbon dioxide, and Krebs cycle is the final common oxidative pathway for carbohydrates, fats, or amino acids through acetyl coenzyme A. Then the third point we have discussed, that is gluconeogenesis. So what is gluconeogenesis? Gluconeogenesis is the synthesis of glucose from non-carbohydrate precursors. The non-carbohydrate precursors, for example, amino acids, glycerol, etc. Then the gluconeogenesis, after gluconeogenesis we have discussed about glycogen. In the glycogen we have discussed about glycogenesis and glycogenolysis. So glycogenesis is the formation of glycogen from glucose, and glycogenolysis is the breakdown of glycogen to glucose. Now today we are going to discuss about hexose 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 monophosphate shunt. So what is hexose monophosphate shunt? The hexose monophosphate pathway, or it is also called as HMP shunt, is also called as pentose phosphate pathway or phosphogluconate pathway. So there are different names for this pathway. This is called as HMP pathway or HMP shunt, or it is also called as pentose phosphate pathway or phosphogluconate pathway. So this is the alternative pathway of glycolysis and TCA cycle for the oxidation of glucose. So in the HMP shunt pathway, this is alternative pathway to the glycolysis and TCA cycle. And in the HMP shunt, it is more of anabolic in nature. So anabolic means it is going to build the different molecules, which can be 3 carbon, 5 carbon, 6 carbon molecules. So it is concerned with the biosynthesis of NADPH and pentosis. So mainly the HMP, hexose monophosphate shunt, it is related to the biosynthesis of NADPH and pentosis. If you remember, we have discussed about glycolysis, then we have discussed about TCA cycle. There we used to calculate what is the total number of ATP in the glycolysis and TCA. We have discussed about the metabolism of glucose leads to how many molecules of ATP. So there we have calculated that total of 32 molecules of ATPs are liberated from metabolism of one glucose unit. So here the HMP shunt pathway, it is concerned with the biosynthesis of NADPH and pentosis. So about 10% of glucose entering in this pathway per day, and the liver and the RBC metabolizes about 30% of glucose by this pathway. And this is the unique multifunctional pathway. The reason is no ATP is directly utilized or produced in this pathway, and it is unique multifunctional pathway since there are several interconvertible substances produced which may proceed in different directions in metabolic reactions. So when we are discussing about the location of the pathway, the enzymes related to hexose monophosphate shunt, they are mainly located in the cytosol. So the tissues such as liver, adipose tissues, adrenal gland, erythrocytes, testes, and lactating mammary gland are highly active in HMP shunt. So most of these tissues are involved in biosynthesis of fatty acids and steroids, which are dependent on the supply of NADPH. So mainly the major location is cytosol, and it is highly active. This pathway is highly active in the tissues such as liver, adipose tissues, then adrenal gland, erythrocytes, testes, lactating mammary gland, and highly active in HMP shunt. These are highly active in HMP shunt. So it is a unique multifunctional pathway because no ATP is directly utilized or produced in HMP shunt, but several other interconvertible substances are produced, which are then proceed to different directions in metabolic reactions, and therefore this is called unique. So mainly HMP shunt is unique pathway. If it is asked why it is unique pathway, it is unique pathway because ATP is not directly utilized or produced, the major important point, but the several other intermediates are produced which proceed in different metabolic reactions, and therefore this is called as unique multifunctional pathway. Multifunctional, why it is called as multifunctional? Because different several interconvertible substances are produced. So different interconvertible substances, it is not like only single one molecule is been produced and then it is proceeding through the different metabolic reactions. So here in front of you, you are able to see the hexose monophosphate shunt. So hexose monophosphate shunt, it starts with glucose 6 phosphate. So basically this hexose monophosphate shunt, it is divided into two parts, or you can say two phases, that is oxidative phase and non-oxidative phase. So if you see this part, the enzyme involved in the first step in the conversion of glucose 6 phosphate to 6 phosphogluconolactone, where glucose 6 phosphate dehydrogenase is the enzyme involved in the presence of the NADP dependent enzyme, that is glucose 6 phosphate to glucose 6 phosphogluconolactone. So this first step, you can say it is the oxidative step, then later is then hydrolyzed by gluconolactone hydrolase. So later the 6 phosphogluconolactone is hydrolyzed by gluconolactone hydrolase to produce 6 phosphogluconate. So you can see here it is producing 6 phosphogluconate. So you can see here you have the C double bond O bond which is being broken over here, and the carboxylate group is free and it forms 6 phosphogluconate. The next reaction involves the synthesis of NADPH, is catalyzed by 6 phosphogluconate dehydrogenase to produce 3 keto 6 phosphogluconate. So it produces 3 keto 6 phosphogluconate, which is then undergoes decarboxylation to give ribulose 5 phosphate. So here you can see the formation of ribulose 5 phosphate. So glucose 6 phosphate dehydrogenase, it regulates the HMP shunt. So this oxidative phase, it is mainly regulated by the enzyme, that is glucose 6 phosphate dehydrogenase. The first reaction catalyzed by glucose 6-phosphate dehydrogenase is most regulatory in the HMP shunt. This enzyme catalyzes an irreversible reaction. So you can see here, this is irreversible reaction. NADPH is competitively inhibits the glucose 6-phosphate dehydrogenase, and it is the ratio of NADPH and NAD plus that ultimately determines the flux of this cycle. So in HMP shunt, the conversion of glucose 6-phosphate to 6-phosphogluconolactone, that is the oxidative phase. The major enzyme which plays an important role is glucose 6-phosphate dehydrogenase, and the enzyme is based on the ratio of NADPH and NADP plus, which ultimately determines the flux of this cycle, and this step is catalyzed by the magnesium. So first phase, that is the oxidative phase. Now next is non-oxidative phase. So if you see here, in the oxidative phase, at the end it is forms ribulose 5-phosphate. Now in the non-oxidative phase, the reactions are concerned with the interconversion of 3, 4 or 5 and 7 carbon monosaccharides. So you can see here the ribulose 5-phosphate, it is 5 carbon chain system. After that, you can see here you have then 6 carbons, then 3 carbon, 7 carbon intermediate molecules are formed, and therefore the non-oxidative phase are concerned with interconversion of 3, 4, 5 and 7 carbon monosaccharides. So the first step in non-oxidative phase, ribulose 5-phosphate is acted upon by the enzyme, that is epimerase, to produce xylulose 5-phosphate, while ribulose 5-phosphate is also acted upon by the another enzyme, that is ketoisomerase. It converts ribulose 5-phosphate to ribose 5-phosphate. So both these are 5 carbon compounds. Then next step is the enzyme transketolase catalyzes the transfer of 2 carbon moiety from xylulose 5-phosphate to ribose 5-phosphate to give 3 carbon glyceraldehyde 3-phosphate and 7 carbon pseudoheptulose 7-phosphate. So again I am repeating, this is the one of the important step in the non-oxidative phase. The enzyme which is involved is transketolase, catalyzes the transfer of 2 carbon moiety from xylulose 5-phosphate. So 2 carbons from xylulose 5-phosphate, they are transferred to ribose 5-phosphate to give. So when 2 carbons are transferred, it forms 3 carbon glyceraldehyde 3-phosphate, and the 7 carbon, when 2 carbons from xylulose 5-phosphate transferred to ribose 5-phosphate, it forms pseudoheptulose 7-phosphate. The important point here is, in this conversion, transketolase is dependent on the coenzyme. You can see here in the blue it is written TPP. So TPP stands for thiamine pyrophosphate. What it is stands for? Thiamine pyrophosphate. And also this step is dependent on the magnesium ions. So this is the very important step where the 2 carbons transferred from cellulose 5-phosphate to ribose 5-phosphate to form 7 carbon pseudoheptulose 7-phosphate and 3 carbon glyceraldehyde 3-phosphate in presence of the coenzyme TPP stands for thiamine pyrophosphate, and also it requires the presence of magnesium ions. Now, in the next step, transaldolase, the enzyme which is involved, it brings about the transfer of 3 carbon fragments from pseudoheptulose 7-phosphate to glyceraldehyde 3-phosphate to give fructose 6-phosphate. So 3 carbons from pseudoheptulose 7-phosphate are transferred to glyceraldehyde 3-phosphate to form fructose 6-phosphate, and the remaining 4 carbons they form erythrose 4-phosphate. So transketolase acts on xylulose. So here in the next step, the transketolase acts on xylulose 5-phosphate and transfers 2 carbon fragments from it to erythrose 4-phosphate to generate fructose 6-phosphate and glyceraldehyde 3-phosphate. So the enzyme involved is transketolase, which transfers the carbons from xylulose 5-phosphate to erythrose 4-phosphate. So transfers 2 carbon fragments from xylulose 5-phosphate to erythrose 4-phosphate to generate fructose 6-phosphate and glyceraldehyde 3-phosphate. Then fructose 6-phosphate and glyceraldehyde 3-phosphate can be further catabolized through glycolysis and citric acid cycle, and glucose may be synthesized from these two compounds. So if you remember, in glycolysis and TCA cycle, the one of the intermediate formed is the glyceraldehyde 3-phosphate. So it enters into glycolysis and moves ahead and generates the energy. So if you see this is the outline of HMP shunt. In the exam, it is expected that you should not study the different structures, but I have mentioned here to understand what is the exact chemistry is happening and how the 2 carbons and shifting of 2 carbons and 3 carbons to form the 7 carbon compound, then 3 carbon compound, 5 carbon compound. So in the exam you can study this cycle where only the names are given and the carbon atoms involved. Here the enzymes are not mentioned, so it is very important that you must mention the enzymes. So again, to review this pathway, it is majorly divided into two: oxidative phase and non-oxidative phase. Oxidative phase, the major enzyme is glucose 6-phosphate dehydrogenase, and the ratio of NADPH and NADP determines the activity of this enzyme, and it forms ribulose 5-phosphate. Then in non-oxidative phase, the ribulose 5-phosphate, by the action of different enzymes, it forms the glyceraldehyde 3-phosphate and fructose 6-phosphate, and then it enters into glycolysis for the generation of energy. So if you see the overall reaction, so I'm not going to discuss, we have already discussed about this. So if you see the overall reaction of HMP shunt, the 6-glucose 6-phosphate, so I'm going back to the cycle. So 6 molecules of glucose 6-phosphate and 12 molecules of NADP plus and water molecule liberates total 6 molecules of carbon dioxide, 12 molecules of NADPH and 5 molecules of glucose 6-phosphate. So what is the significance of HMP shunt pathway? So you can see over here, what is the major role of HMP shunt pathway? It forms two products, that is pentosis and NADPH. So you can see here in the cycle, the major You can see here these are the 5-membered carbons. So 5-membered carbons. So basically it forms pentoses, and it generates the NADPH. So here this is one of the step. In the first step, glucose 6-phosphate to 6-phosphogluconolactone, it generates NADPH. Then here in the 6-phosphogluconate to ribulose 5-phosphate, it generates NADPH. So these are the important steps. So HMP shunt is unique in generating two important products, that is pentoses and NADPH. The in HMP shunt, hexoses are converted into pentoses, the most important being the ribose 5-phosphate, which is being produced at the end of the oxidative phase, and then it enters into non-oxidative phase. And the pentoses which are formed and its derivatives, they are useful for the synthesis of nucleic acids like DNA and RNA. Also, it is used for the synthesis of nucleotides such as ATP, then NAD, FAD, and coenzyme A. So the significance of HMP shunt pathway is it majorly involves in the formation of two important products, that is pentoses and NADPH. So the pentoses, they are majorly useful in the synthesis of nucleic acids like RNA and DNA, and also many nucleotides such as ATP, NAD, FAD, and coenzyme A. Skeletal muscles are also capable of synthesizing pentoses, although only the first few enzymes of HMP shunt are active, and therefore it appears that complete pathway of HMP shunt may not be required for the synthesis of pentoses. Then another important part is it forms the NADPH. So what is the role or what is the use of NADPH? As we all are aware that the NADPH is required for the reductive biosynthesis of fatty acids and steroids, and hence HMP shunt is more active in the tissues concerned with lipogenesis. Example, adipose tissues, liver, etc. Then NADPH is also used in the synthesis of amino acids such as uh, glutamate dehydrogenase is the enzyme involved. Uh, so they are also used in the synthesis of amino acids, and the enzyme involved is glutamate dehydrogenase. So first important point is related to NADPH. It is required for the biosynthesis of fatty acids and steroids, and hence it is more active in the tissues concerned with lipogenesis. Example, it is adipose tissues and liver. Then second important point is it is used for the synthesis of amino acids involving the enzyme, that is glutamate dehydrogenase. Then third important point is it act as free radical scavenging. So there is continuous production of hydrogen peroxide in the living cell, which can chemically damage the lipids, proteins, and DNA. So this is prevented by antioxidant reactions, which is involving NADPH. So it is very important. Then next is a glutathione mediated reduction of H2O2 is involved. Then it also act as the free radicals. Superoxides, hydrogen peroxides are continuously produced in all cells. So NADPH is playing very important role in inhibiting the action of these free radicals. So these free radicals, they can destroy the proteins, fatty acids, biomolecules, and therefore they are inactivated. These free radicals are inactivated by the enzyme systems containing the glutathione, then SOD, POD, etc. So reduced glutathione is regenerated with the help of NADH. The next is detoxification of drugs and foreign compounds. So microsomal cytochrome P450 system, it brings the detoxification of drugs and foreign compounds by hydroxylation reaction involving NADPH. Then another important use is it act as phagocytosis. It acts in phagocytosis. So what is phagocytosis? It is the engulfment of foreign particles, including microorganisms, carried out by white blood cells. So this process requires supply of NADPH. Then the next special function is it prevents the it preserves the integrity of RBC membrane. So special function of NADPH in RBC. So it produced NADPH produced in erythrocytes has special function to perform. It maintains the concentration of reduced glutathione, which is essentially required to preserve the integrity of RBC membrane. So if you see majorly the important points related to what we can say pentosis and the NADPH, because majorly HMP shunt is involved in the synthesis of pentoses and NADPH. So pentoses, it is majorly involved in the synthesis of nucleic acids like DNA and RNA, and also different nucleotides such as ATP, NAD, FAD, and coenzyme A. And related to NADPH, the 5 to 6 are the key points. First point, important point is it involved in the biosynthesis of fatty acids and steroids. Second important point, it is involved in the synthesis of amino acids. Then third is it act as antioxidant by inhibiting the free radicals formed in different reactions. Then next important point is uh, it helps in the detoxification of drugs and foreign compounds by hydroxylation reaction. Then another important point is phagocytosis. It helps in phagocytosis. So it, phagocytosis is the engulfment of foreign particles, including microorganisms, carried out by white blood cells, and this process requires the NADPH. Then the last important function is it maintains the integrity of RBC membrane. So these points we have discussed. Now next part in the HMP shunt pathway is glucose 6 phosphate dehydrogenase deficiency. So as you have seen in the cycle, the enzyme which is involved in the first step, that is glucose 6 phosphate dehydrogenase. So it is also called as G6PD deficiency. So we are going to discuss about that. So G6PD deficiency or glucose 6 phosphate dehydrogenase deficiency is an inherited sex linked trait. So you can see here it is inherited sex linked trait, and the gene of G6PD is on the X chromosome, and hence only the male they are affected in this case. This is not observed in case of females. So G6PD deficiency is regarded as the most common genetic defect, as about 400 million people worldwide carry mutated gene. So decrease activity of this enzyme impairs the synthesis of NADPH in the RBCs, and because of which this results in accumulation of methemoglobin and peroxides in erythrocytes, leading to hemolysis. So the enzyme involved here is very important, that is glucose 6 phosphate dehydrogenase. The deficiency of glucose 6 phosphate dehydrogenase leads to hemolysis. The deficiency is manifested only when it is exposed to certain drugs or toxins. Example, intake of antimalarial drugs like thymoquine and ingestion of the fava beans or sulfa drugs. It also precipitates the hemolysis, and the situation is called as hemolytic anemia. So the clinical manifestation in most patients with G6PD deficiency do not usually exhibit clinical symptoms, but some of them develop the hemolytic anemia, and if they administered the oxidant drugs or exposed to severe infections. The drugs like antimalarial drugs like primaquine, then acetanilide, that is antipyretic drug, then sulfamethoxazole antibiotic, so sulfa containing drugs, then the ingestion of fava beans, they produce hemolytic jaundice in these patients. Severe infections results in the generation of free radicals which can enter the RBC and leads to hemolysis. So hemolysis is basically because of severe infection which leads to formation of free radicals, and those radicals then they enter into RBC and causes hemolysis due to decrease NADPH and reduce glutathione hormone. So G6PD deficiency and resistance to malaria. So it is interesting to note G6PD deficiency is associated with resistance to malaria, and this is explained from the fact that parasite that causes malaria are dependent on the parasites, they are dependent on HMP shunt and reduced glutathione for their optimum growth in RBC. So it is observed that G6PD deficiency and it is resistance to malaria. So how it is being diagnosed? The diagnosis, the biochemical diagnosis can be done by detecting the reduced activity of G6PD in RBC. The management of deficiency, G6PD deficiency includes avoiding oxidative stress and symptomatic treatment of hemolysis. So that was related to glucose 6 phosphate dehydrogenase deficiency. So this part we have discussed. Now the next part is blood glucose regulations. So as we all are aware about the blood glucose regulations, the normal plasma glucose level, if you see here, it is indicated, then factors maintaining blood glucose, postprandial regulations. So what is glucose homeostasis? It is the maintenance of blood glucose level within the normal range. So what is the normal range? It is 70 to 100 milligram per deciliter. Then postprandial blood glucose level is 120 to 140 milligram per deciliter. So above and below of this level, it is considered as abnormal. If it is a level above than the normal, then it is called as hyperglycemia, and if it is below than the level, it is called as hypoglycemia. So in the carbohydrate metabolism, we have discussed about uh, glycogenolysis, then gluconeogenesis. So you can see here in this diagram, these are the factors which maintain the blood glucose level. So what is observed here, in case of normal situation, the blood glucose level is 70 to 110 milligram per deciliter. So mainly the supply of the glucose is through the diet after digestion and absorption. So here, if you see on the left side, it is indicated glycogenolysis. So liver glycogen, they are utilized. So source of energy during early fasting. So whenever there is a fasting, so here the glucose supplying through the diet has been reduced, and therefore the glycogen is being metabolized, or glycogen is being broken down to glucose, and that is being used to maintain the normal blood glucose level. In case prolonged starvation, which is more than 18 hours, then in that situation gluconeogenesis occurs, and it is mainly from the amino acids. So again, in that situation, the glucose supply through the diet is not possible, and therefore more than 18 hours, then it is gluconeogenesis. If it is 12 to 18 hours, then it is glycogenolysis, and then the blood glucose is utilized for different purposes, that is for the oxidation reactions, then glycogenesis and lipogenesis. So here it is explained the homeostasis of blood glucose. Whenever blood glucose level decreases, so pancreas, it activates the alpha cells to release the hormone, that is glucagon, and then glucagon carry out the glycogenolysis in the liver, and glucose is released to the blood, and it reaches to the normal blood glucose level. If there is high blood glucose, then the pancreas, they secretes, beta cells secretes the insulin. Then insulin, in the peripheral tissue cells, takes the glucose from the blood, so uh, it brings the blood glucose to the normal level. So factors involving the homeostasis of blood glucose, so sorry, metabolic process, hormones and renal mechanisms are involved to maintain the normal blood glucose level. So as discussed, the blood sugar tends to increase. As the blood glucose tends to increase, then it increases the secretion of insulin, glycogenesis is accelerated, and utilization of glucose by tissues is increased. And as the blood glucose sugar tends to decrease, then it decreases the secretion of insulin, it decreases the ratio of insulin to glucocorticoid and GH hormones, and it increases the production of glucose mainly by gluconeogenesis and glycogenolysis, and decreases the utilization of glucose by the tissues. So, here today we have discussed about HMP shunt pathway, then G6PD deficiency, that is glucose 6 phosphate dehydrogenase deficiency, and how the glucose is maintained, how the carbohydrate metabolism plays an important role in the maintaining the normal blood glucose level in the body. So the important questions which are asked on this topic is, the major question which is asked is, describe the HMP shunt pathway, give the significance of HMP shunt. So when it is asked about the significance of HMP shunt, it is really important that student must write the importance of pentosis, then importance of NADPH, and majorly the two parts the question can be asked. First is, discuss the HMP shunt pathway. That is the one major question. And the another major question is, discuss the significance of HMP shunt pathway and glucose 6 phosphate dehydrogenase deficiency. So the deficiency, then you have to discuss about clinical manifestations in G6PD deficiency, then G6PD deficiency and resistance to malaria and its biochemical diagnosis. So we'll stop here. Thank you. If you have any doubts, you can be in touch with me.